Okay, ladies and gentlemen, I want to thank everybody for showing up today. We're having the MS Thesis Defense for Mr. Orville Baldos. He will be discussing his thesis work of the assessment of hydroplanting techniques and herbicide tolerance of 2 native Hawaiian ground covers for roadside revegetation potential. A little background on Orville: he received a BS in landscape horticulture from the University of the Philippines, Los Baños in 2002. He did various other landscape-related activities and jobs in the Philippines up to about 2005 when he became a teaching assistant for the department. Did that for a year. In 2006, he joined my program to start his work on trying to look at the use of native Hawaiian plants for roadside revegetation. The grant that he's being supported on is designed to develop protocols for the establishment of these plant materials, and that's what he's been busily doing for the last couple years. As you can see here, we have kind of a studio set up today. Part of the grant's deliverables is stakeholder outreach. So we're going to use this opportunity to record his presentation for use as an internet presentation. I invite the other graduate students that are here and faculty members. Should you have guest speakers or anybody else and you would like to have a similar setup, you know, we got different backdrops and we can set this up. The idea here is to have a good rendition of the speaker. We then take the PowerPoint presentations and lay them over the person's head in a video editing program. So we have, and then we can compress it for use on the web. So without any further ado, I'd like to invite Orville Baldos to take the stage. I'm not going to kiss him or anything, but good luck, Orville.
Thanks. All right. Thanks all for coming. But before I proceed in detail discussing the studies that were included in the thesis, I'll just give you a brief outline of this afternoon's presentation. So first I'll give an overview of roadside revegetation, what's currently being done here in Hawaii, and then proceed to thesis objective and then the species information about, because I have 2 species. So I'll just give a brief background about the 2 species I studied. And after that, we'll proceed in detail discussing the studies that were involved for the thesis. So I divided it into 2 portions. I have the hydroplanting techniques and then the herbicide tolerance portion. So after discussing it in detail, I'll give a short summary and some topics for future studies. So why do we do roadside revegetation? Revegetation is basically done in order to mitigate the impacts of roads and road construction. They're primarily done in order to promote motorist safety, to mitigate runoff and pollution, and provide roadside aesthetics. So conventional roadside revegetation utilizes native species because they are cheap, readily available, and easy to establish on disturbed sites. Although they possess these desirable characteristics, they may fail due to site constraints, and they may also proliferate and become noxious. In recent years, the focus on revegetation has been on native plants because of risk of non-natives, potential risk with non-natives, and there's also a growing awareness about conserving local biodiversity. And for the roadside people, the people who maintain the roadsides, they wanted to reduce maintenance costs. So why use native species for roadsides? According to Roadside and Hunt, roadside revegetation with native species is the most environmentally sound and aesthetically pleasing way of establishing vegetation. Native species are adaptable to local conditions. They preserve local biodiversity, and they also, by planting native species on the roadside, you prevent noxious weeds from growing in these areas. Thereby, you stop the spread of invasive weeds. So instead of having that space for non-native plants, you have the native plants in the roadsides. So in addition to environment and aesthetics, roadside revegetation with native species can also lower maintenance costs. So in Hawaii, revegetation utilizing native Hawaiian plants is a new initiative. The Hawaii Department of Transportation is leading the way to develop native alternatives to Bermuda grass. Because Bermuda grass is their current species that they're using right now in revegetation. So they wanted to include more native species in their roadside landscaping projects. So in the past years, they have successfully integrated some native shrubs, but a lot of research is still needed, particularly in the establishment of ground covers. So for this thesis, we have our main objective is basically to evaluate and propagation and selective weed control for two species, namely Fimbristylis cymosa and Sporobolus virginicus. Fimbristylis cymosa is Mao-aki-aki in Hawaiian. It is a tooth-growing perennial coastal sedge with potential for revegetation, landscaping, and riparian restoration. In Hawaii, it is found in all main islands except for Koholave. This species, since this one is a coastal species, it is salt, wind, and drought tolerant. And currently, constraints meaning constraints to roadside use is that right now there's no alternative to outplanting. So what this thesis aims is that we wanted to find alternatives to outplanting of plugs. And then also there's no weed control protocol for establishment. So weed control during establishment is essential because the seedlings are pretty sensitive, or they're vulnerable to weed invasion, so you want to control the weeds during that time. So the next species is Sporobolus virginicus, or aki-aki. Aki-aki is a perennial coastal, rhizomatous coastal grass with potential use as a low-maintenance industrial turf. This species is found in subtropical and tropical areas of the world, and here in Hawaii it is found in all main islands. This species is salt and drought tolerant, and currently we wanted to find alternatives again for outplanting, and there's currently no weed control protocol for this species. So this thesis is basically divided into two main focus areas. We have the propagation focus and the weed control focus. So for propagation, we wanted to develop alternatives to outplanting. So we wanted to develop hydroplanting techniques for these two species. For the weed control focus, we wanted to evaluate tolerance of these two species to selective herbicides labeled for roadside right-of-way use. Because by having this data over here, we can use certain herbicides during establishment. So we want to control, or we want to develop a weed control protocol using herbicides. So this is the breakdown of the thesis studies. So we have under hydroplanting techniques, we have two areas. We have planting and then planting plus weed control. For selective weed control, we have it divided into 4 areas. We have a pre-emergence, and then we have control for post-emergence control for different types of weeds. So same is found here in the second species, which is Sporobolus virginicus, but certain areas are left blank, because some are already included and some are already done. So for the propagation focus, we wanted to develop hydroplanting techniques by utilizing seeds or seedlings for Fimbristylis cymosa, and then stem cuttings for Sporobolus virginicus. So for Fimbristylis cymosa, we had field assessment of hydroplanting techniques. So this experiment had 3 treatments over here. So we have hand-sown seedlings capped with hydromulch. We have hydroplanted seedlings, and then we had hydroseeding. So this study was conducted at the H1 University Avenue off-ramp Cloverleaf. As you can see here in this recent satellite photo, you can see where our experiment was conducted. So this square over here. So you can see the individual plots. So prior to field assessment, we conducted seed counts and germination tests. So for the seed count, we found that there was around 298 seeds per 0.02 grams, or around 15,000 seeds per gram. We also did germination test, and it was around 91%, so it's pretty good. So for the treatments in detail, we have hand-sown seedlings capped with hydromulch. We had this density over here, and then we mulched it with 4,400 kilograms per hectare. So for this treatment, what we did was we grew the seedlings 2 months prior to the experiment, or prior to treatment application. We planted them by hand on the prepared planting surface, and then we covered it with hydromulch. So this is how it looks like. For the second treatment, which is hydroplanted seedlings, we had the same amount of seeds or densities, planting density, and same mulch. The difference is that we mixed the seedlings inside the hydromulching machine, and then we sprayed it onto the surface. The third and last treatment for this study was hydroseeding. So we used approximately double the amount of planting material, and then half of the mulch. So for this treatment, we mixed the seed and mulch in the hydromulcher, and then we sprayed it onto the planting surface. So each area here, which covered approximately 3.1 square meters, we used 0.1 grams of seed. So you can see here to scale with the finger. So we use that small amount of seed. So during the first 6 months after planting, we collected plant counts for the first 2 months and percent visual cover for the first 6 months. So these two measurements were collected in a 1 by 1 square foot sample area, which is around 0.09 square meters. And for plant counts, we manually counted the number of seedlings that came up for the first 2 months. And then for visual cover, we took photographs of the sample area, and then we superimposed square grids in Adobe Photoshop, and then just counted the squares manually as an estimate of cover. So plant count results and percent survival for the first 2 months. You can see here that the hydroseeded treatment consistently attained high plant counts per sample area, followed by the hand-sown seedlings, and then the lowest was attained, or the lowest plant densities were observed in the hydroplanted treatment. So for percent survival, this was actually the plant counts attained during the second month, and then the plant counts attained during planting. So based on the densities that were planted during planting. So for hydroseeding, we had 62% survival. And then for hand sowing, we have 41%, and the least survival was observed in the hydroplanting treatment. So this is the monthly visual cover results over the 6-month period. So as you can see here that after the first month, you can see that the hydroseeding treatment had the highest, consistently attained the highest percent visual cover, followed by the hand-sown or hand-dropped seedlings. And then the lowest was observed in the hydroplanted treatment. So this is a representative sample, or a representative photograph of a plot. So A is hand-sown seedlings covered with hydromulch, B is hydroseeding, and C is hydroplanted seedlings. So you can see here during the first month, there is not much cover visually when you compare each treatment. But as you increase the time, or as time passes, you can see here that visually the hydroseeding treatment had more densities than the hand-sown treatment, primarily because it had higher seeding, or higher amounts of seeds used. So after 6 months, there's not much any difference between the two, A and B. So based on this study, hydroseeding and hand-sowing hydromulching treatment is feasible, but hydroseeding seems to be a more practical method of planting because seeds are, they don't, and when you compare hand sowing, because you need to grow seedlings months prior to planting. So for hydroseeding, it doesn't require that time and resources. And hydroplanting of seedlings is not advisable because when you mix or when you put the seedlings inside the hydromulcher, it cuts the seedlings or it damages it. So you have little to almost no seedlings or plants that are generated from this planting method. So now we know how to hydromulch Fimbristylis cymosa. Let us now proceed to the next species, which is Sporobolus virginicus. So for this study, I evaluated stem cuttings for hydromulch planting. So I divided it into two sub-studies. So I wanted to know first if apical or basal cuttings would be, which ones or which part of the stem can be used, or which are the best for use in this next study. So I did this first before doing the hydromulch planting, rooting response under hydromulch planting conditions. So for the first experiment, so we have apical versus basal cuttings. So we have two treatment factors basically. We have plant part and we have soaking times. For plant part, I have it divided into apical cuttings and basal cuttings. Apical cuttings meaning it's leafy and it's the top 10, it has 10 nodes and it's around 20 cm in length. Basal cuttings are generally leafless because they're below the canopy level and they have 3 nodes in them. And then we have soaking time, so we have a no-soak treatment, a 5-second dip, and a 3-hour soak. So the soaking times, the cuttings were soaked in soaking solution containing 500 ppm IBA and 250 ppm NAA. This is basically a 1 to 20 dilution of Dip and Grow, which is a commercially available rooting solution. So for this study, we harvested the cuttings, separated them into apical and basal portions, and then we soaked them in their respective soaking times and planted them in pots filled with moistened potting mix. So these pots were allowed to root for around 21 days inside the shade house with continuous misting, and then after 21 days we placed it outside under field conditions, irrigated field conditions, to allow further rooting. So 35 days after planting, we collected percent rooting, visual rooting scores, which range from 1 to 5. So 1 means that the cutting is dead, and this is basically what the visual rooting score would look like. So a rooting score of 5 would mean that you have very profuse rooting of cuttings. And then besides visual rooting scores, we also collected length of longest root. So you can see here a photo of the cuttings 35 days after planting. Basically, you see here that as you increase the soaking time, you generally have more cuttings that rooted for apical cuttings. And then for basal cuttings, there's not much any difference visually. And you can see when you compare apical and then basal cuttings, apical cuttings are clear. You can see that they're better performing, or they root better than the basal cuttings. So for percent rooting, there was a significant interaction with soaking time and plant part, and the highest values were attained in the 3-hour soak, followed by the 5-second dip. For root density scores, you see the same trend. You have the highest rooting score in the 3-hour soak, and followed by the 5-second dip. And then this one is actually apical cuttings. And then basal cuttings was not significantly different from each other, regardless of soaking time. For length of longest root, you also see the same trend, with the apical cuttings having the highest, or having the longest root length, and then the basal cuttings, regardless of soaking, was not significantly different. So based on the results of this study, leafy apical cuttings are the ones that have the best rooting. So the next step was to evaluate it under hydromulched planting conditions. So for this study, we have 10-node apical cuttings with all the leaves intact, and then we have 4 treatments, basically soaking time. So we have untreated cuttings, we have a 5-second dip, a 4-hour soak, and a 24-hour soak in rooting solution that was described earlier. It's the same rooting solution that we described earlier. So it's 1 to 20 dilution of dip and grow. So for this study, we separated the cuttings. We harvested cuttings, soaked them in rooting solution, planted them in trays, and then we covered it with hydromulch, as you can see here in this video. And after covering it with hydromulch, we placed it in the field and allowed to root for 45 days. So we collected percent rooting scores, and number of new green shoots per cutting 45 days after planting. So here is a representative sample of each treatment. So you can see that as you increase soaking times, you have generally an increase in rooting of cuttings. So in terms of percent rooting, you see some increase with longer soaking times. The ones soaked for 24 hours had the highest percent rooting. Same trend was observed in the rooting score, with the 24-hour soak having the highest value, and then the others are not significantly different if you compare it with the untreated cuttings. And then for green shoots, you have more green shoots per cutting with longer soaking times. So for this study, we can the best rooting was observed in cutting soaked for 24 hours. And this study also exhibited or showed that hydroplanting establishment using cuttings is feasible, and it can be an alternative to rooted plugs, and it can bypass nursery operations. So you can establish it on site. So now we know this area over here. Let us now proceed to the selective weed control of Fimbristylis cymosa. So for the weed control focus of the study, we evaluated tolerance of the 2 species to selective herbicides labeled for roadside right-of-way use. So for Fimbristylis cymosa, we have 3 studies in the weed control focus. So we evaluated first pre-emergence herbicides alone, and then we evaluated post-emergence herbicides alone. And then what we got from these 2 experiments led to the 3rd experiment, which was timing of pre- and post-emergence application. So for the pre-emergence screening of for Fimbristylis cymosa, we have 2 herbicides, pre-emergence herbicides. Pre-emergence herbicides, by the way, are herbicides that you apply to control germinating weed seeds. It doesn't control for emerged weeds. So we have 2 herbicides. We have Oxadiazon and then oryzalin, and then a combination of both. We have the label rate and twice the label rate of each. And then we also have an untreated control. So this study was one of the first experiments conducted for this thesis. So this was conducted at the NRCS Plant Material Center in Molokai. And what we did was we grew plugs, planted them in rows, and then sprayed the pre-emergence herbicides a day after planting. 15 days after spraying, we fertilized the plots. 40 days after planting, we weeded the plots, and then reapplied 44 days after planting. So basically 4 days after weeding, we sprayed the plots again with the pre-emergence herbicides. 211 days after the first spraying, we collected visual plant vigor ratings, seed head counts of 2 representative plants. So you can see here at 211 days, the untreated plots were weedy, and then the ones that were sprayed with pre-emergence herbicides had little to almost no weeds. You also notice here that when you apply double the label rate of oxadiazone, you see a stunting of the plant. But for oryzalin, when you double it, you don't see an obvious stunting of plants. With the combination of the two, oxadiazone and oryzalin, at the low or at the label rate, you don't see a stunting. But when you have it twice the label rate, you see some stunting of the plants. So in terms of vigor rating, you can see that the highest vigor rating was observed in the oryzalin treated plants, and then the lowest was in the untreated plants. The reason why the untreated plants was the lowest because the plots were weedy, and the weediness actually affected the vigor of the plant. In terms of seed head count, there was no significant difference in terms of seed head count for each of the treatments. So based on the results of this study, we can use oryzalin and oxadiazone for transplanted plugs, so for pre-emergence weed control of transplanted plugs. So now we know this part of the study. Let us now proceed to the post-emergence study, which was divided into 3 areas. Again, so we have the first study or the first experiment conducted for this area of the thesis was this one. So we evaluated Fluazifop-p-butyl and triclopyr. Fluazifop-p-butyl is a post-emergence herbicide that controls grassy weeds. And then triclopyr is a post-emergence herbicide that controls broadleaf weeds. So we sprayed them on plants of different age classes to see if there's any injury. So the second study was post-emergence broadleaf herbicide screening for field-established plants. And then the third one was tolerance of field-grown plants to 2 rates of aminopyralid. So for the first study, which is Fluazifop-p-butyl and triclopyr on different age classes, we had 2 treatment factors. So we have plant age, and then we have the different herbicides. So for plant age, we have 224-day-old plants, 98, and then 43-day-old plants. For the herbicides, for Fluazifop-p-butyl, we have high and low label rates plus CO. CO is crop oil. And then for triclopyr, we have high and low label rates again, plus crop oil. So we have a crop oil treatment and then water. The reason for this is that we wanted to know if there's any effect of crop oil. So this study was conducted at the Magoon Research Facility in Upper Manoa campus. And what we did was we sowed the seeds. So in order to get seeds or plants of different ages, we sowed the seeds at different periods of time. Then once we established these seedlings, we sprayed the treatments. So 28 days after spraying, we collected foliar injury ratings ranging from 0, meaning no visual injury, and 100, meaning complete plant death. So in terms of injury, there was no significant interaction with age and herbicide treatment. So the response was not affected by age, so we just pulled the injury ratings within the ages. So you can see here that triclopyr was very injurious to the plant, while Fluazifop-p-butyl had 0 injury, and the same was observed in the control and crop oil treatments. So 28 days after spraying, this is like a sample of the treatments, the different treatments, and then you have the different ages of the plants. So you can see here that the triclopyr treated plants were dead, while Fluazifop-p-butyl at the low and high rates were comparable to the control visually. So for this study, we can use Fluazifop-p-butyl safely for seedlings at least 48 days old. And then for triclopyr, we cannot use it because it killed the plant. So we need to find other broadleaf alternatives for Fimbristylis cymosa. So we know how to control the grass. Now we need further studies for broadleaf. So this led to the next study, which was post-emergence broadleaf screening in field established plants. So we have 5 treatments over here. We have aminopyralid, which is milestone VM. We have a 4 herbicide mix called Power Zone, and then we have another herbicide mix called Speed Zone. The difference between the two is that instead of MCPA, for Speed Zone, instead of MCPA, it was replaced with 2,4-D. And then sulfosulfuron is another herbicide that controls for sedges, but it also has some control for broadleaf weeds. So you notice here that the aminopyralid was overapplied in this experiment. We had it confused when we got the bottles. So we had milestone VM and milestone VM plus, so because it looked the same, so we have it overapplied. So this study was conducted at the Waimanalo Research Station. So a year prior to treatment application, we flushed the weeds subsequently, and then we rotavated or tilled the field, measured some rows, and then we also did some liming and fertilizer applications prior to planting. And then we installed overhead irrigation, and prior to planting, we weeded the plots. So this is the area here. So a year prior to post-emergence screening, we produced plugs, planted them in rows, and then we sprayed the rows with the different herbicides 70 days after planting. So 36 days after spraying, we collected vigor, injury, and green color ratings. And then we also collected percent mortality. So you can see here that 36 days after spraying, you can see that all of the herbicides were injurious to the sedge. The aminopyralid, which was overapplied, had overall yellowing of the plants, while Power Zone and Speed Zone had necrotic damage, which was light brown in color. And then sulfosulfuron, the sedges were stunted, severely stunted, and it had a distinct dark black necrosis on the leaves. So in terms of percent vigor, you can see, based on the picture previously shown, you can see that all of the herbicides had lower, significantly lower percent vigor. For percent injury, the aminopyralid, Power Zone, and Speed Zone had higher injury ratings compared to the sulfosulfuron and untreated plants. And then for green color, you can see that sulfosulfuron was not significantly different from the untreated plant, whereas the aminopyralid, Power Zone, and Speed Zone treatments were significantly lower in green color because they had high injuries. In terms of mortality, aminopyralid, Power Zone, and Speed Zone had mortalities recorded in them, while sulfosulfuron had no mortality. So based on the data that was collected for this study, we ranked the herbicides from most to least injurious. So the most injurious was Speed Zone, followed by Power Zone, and then aminopyralid and sulfosulfuron. So based on this ranking, based on this ranking and the data presented, you should only spray Speed Zone, Power Zone, and sulfosulfuron as a directed spray, because if you spray it on the sedge, it will injure it badly. So for aminopyralid, since we overapplied it 25x, we need to conduct a further study to confirm if this one is safe for the sedge. So we had a follow-up study in which we applied the right label rates of aminopyralid. So we have a high and then a low, low and a high rate, and we used the control plots from the previous post-emergence study. So we collected foliar injury and green color ratings 35 days after spraying the different treatments. So you can see here, 35 days after treatment, there's only a slight difference in terms of color, I guess, when you compare plants, untreated plants with the aminopyralid treated plants. When you look at the quantitative data, vigor was not significantly different from the treatments. And then for injury, there was a significant difference, but the injury levels were slight and they were acceptable enough. Green color ratings also had a significantly, the aminopyralid treatments had a significantly lower green color ratings, but it was still above 90%, so it still had acceptable injury ratings. So based on the results of this study, we can use high and low label rates of aminopyralid to control broadleaf weeds. So you can see here, we were able to control Lucina, Leucocephala, with spray of aminopyralid. Then we can also control Portulaca oleracea on the sedges. So we can selectively remove the Portulaca on the sedge. So now we know how to control the broadleaves in this species, and then we can spot spray the sedge. We now combine the two experiments. So what we got from this one and then this one led to this study over here, which is we wanted to know how early can you apply pre and post-emergence herbicides on hydroseeded F. cymosa. So yeah, so how early can you apply pre-emergence and how early can you apply post-emergence? So we have the different treatments over here. We have two pre-emergence herbicides, Oxadiazon as a wettable powder, oryzalin as a spray, and then we have a pre-A, which is Oxadiazon as a wettable powder, and then a post-mix of aminopyralid and Fluazifop-p-butyl. So this one controls broadleaf weeds, and then this one controls grassy weeds. And then for pre-B plus post, we have Oxadiazon as a granular, plus a mix of aminopyralid and Fluazifop-p-butyl. We also have a post-only treatment of aminopyralid and Fluazifop-p-butyl. So this is how the treatments look like several days. This is how we applied the treatments in terms of days after hydroseeding. So we applied pre-emergence 7 days, pre-emergence 1, which is a wettable powder, and oryzalin 7 days after hydroseeding. And then 14 days after, we applied the other treatment, which had the same pre-emergence herbicides. And then 28 days after, we had a pre-A plus post, and then a post-only treatment. 42 days after hydroseeding, we sprayed pre-B, which is Oxadiazon as a granular, and then we have the post-only treatment of aminopyralid and Fluazifop-p-butyl. And then at 56 days, we only sprayed post-emergence. So before we applied the treatments in the plots, we hydroseeded the whole field with the sedge. And then 7 and 14 days after, we applied pre-emergence, and then we applied post-emergence with or without pre-emergence at 28, 42, and 56 days after hydroseeding. So plant counts were collected at 92 days, and this is how the plot looks like 92 days after. So you can see that the pre-emergence Oxadiazon had some weed control, actually good weed control here, compared to untreated, but even though it had good weed control, it also controlled the sedge, so no sedge seeds were alive. For oryzalin, another pre-emergence herbicide, you have some weed control also with applications at 7 and 14 days, but you have some seedlings popping up, but they were smaller than the untreated plants. For the post-emergence treatments, so this one is pre-emergence plus post, and then as a wettable powder, and pre-emergence plus post-asagranular. So you can see here that it had good weed control and also controlled the sedge when you apply a pre-emergence as a wettable powder and a post-emergence at 28 days. 42 days, when you apply granular and post, you see some seedlings, but the granular application actually injured some of the seedlings here. So you still have a reduced number of seeds per square meter. The post-emergence only treatments had good control of some control of weeds compared to untreated plots, but you see here that these plots are weedy because the plots were actually invaded by spurges, and aminopyralid doesn't control for spurges. So you can see here that the addition of pre-emergence plus post killed all of what's in the plot, and pre and post applications as a granular, you still see some seedlings here, but the seedlings are badly damaged by the pre-emergence application. And then when you apply post-emergence only at 28, 42, and 56 days, you can see that it had comparable number of plants with untreated. So plant counts 92 days after seeding, hydroseeding, revealed that the ones sprayed with post-emergence only had high plant counts compared to the ones sprayed with the pre-emergence. So based on the results of this study, we can use oxadiazon and oryzalin for, we cannot use oxadiazon and oryzalin for hydroseeded plants less than 42 days old. We can use it in plugs, but not for hydroseeded F. cymosa. For aminopyralid and Fluazifop-p-butyl, we can control broadleaf and grassy weeds 28 days after hydroseeding, as early as 28 days. And you saw in the previous photos, and you can see here in this photo here, aminopyralid doesn't control for spurges, so you have an invasion of spurges after you sprayed the post-emergence. The reason why we didn't use oryzalin for this experiment is that when we mixed oryzalin with aminopyralid and Fluazifop-p-butyl, we had an incompatible solution, so it precipitated, so cannot be used. So now that we know this part of the study, let us now go to the next species, which are the weed control for the next species. So we have Sporobolus Virginicus herbicide tolerance. So this is basically divided into experiments which evaluated pre-emergence, and then we have post-emergence on established plants. So for the pre-emergence study, we have 4 herbicides here. We have high and low rates of oxadiazone, high and low rates of trifluoralin plus isoxaben, and high and low rates of dithiophyr, high and low rates of oxyfluorfen, and then we added another treatment, table salt, because we thought that maybe we can control some weeds with application of salt, since Sporobolus virginicus is tolerant to salt. So for this study, we produced plugs a year before. We planted the plugs in rows, and then we applied herbicides as a granular or as a spray, depending on the herbicides. 70 days after spraying, we weeded the plots, we removed the biomass, above ground biomass, and then we reapplied the pre-emergence herbicides. 38 days after the second application of herbicides, we rated injury, and we got some time weeding data, and we collected above ground biomass. So you can see here 38 days after the second application of herbicides, you can see that oxadiazone and oxyfluorfen had excellent weed control compared to untreated and table salt. The ones with trifluoralin plus isoxaben had some control of weeds, but it wasn't as good as the 2 previous herbicides discussed or shown. So in terms of foliar injury, you can see here that there was no significant difference within with the treatment, with the different treatments in terms of foliar injury, because the foliar injury actually ranged from 0 to 1, so it was really slight. In terms of time weeding, oxyfluorfen and oxadiazon had the shortest weeding times. So you have less than 5 minutes to weed 8.8 square meters, whereas for untreated, it took us 25 minutes to weed it. Table salt also was comparable to untreated, and it took us like 15 minutes to weed it. Oxadiazon and oxyfluorfen also had the highest above ground biomass compared to the other herbicides, and salt had the lowest above ground biomass. So based on this study, we can use oxadiazon and oxyfluorfen because it had the shortest weeding times, it had the greatest control of weeds, and it also had the shortest weeding times, and it had the highest biomass. So there wasn't any, or it had the least growth inhibition, or in terms of when you compare it with other herbicides. So now we know the pre-emergence study. Let us now proceed to the post-emergence study for Sporobolus. So we have 5 post-emergence treatments. We have aminopyralid, and then we have triclopyr, we have PowerZone, and sulfosulfuron. Notice here that we had an over-application of aminopyralid because this experiment was actually done at the same time as the Fimbristylis. So yeah, we had it over-applied at that time. So this study was conducted at the Waimanalo Research Station, and we produced plugs, and we planted them and fertilized them. We applied oxadiazon, a pre-emergence herbicide from the previous study, because we can use it safely for this grass. Then after fertilizer application and pre-emergence herbicide application, we applied the post-emergence herbicides at 98 and 133 days after planting. So 28 days after the second application of herbicides, we collected vigor, injury, and green color ratings. We also collected above ground biomass of 3 representative plants. So you notice here that injury ratings, 28 days after the second application, you can see that aminopyralid and triclopyr had the highest injury rating, followed by PowerZone, and then the lowest injury ratings were observed in the sulfosulfuron treatment. Green color ratings were high in the sulfosulfuron because it had low injury, and then PowerZone had the next highest, and then the lowest green color ratings were observed in the aminopyralid and triclopyr treatment. Vigor was also had the same trend, with sulfosulfuron having the highest among the herbicides tested, followed by PowerZone, and then the lowest was triclopyr and aminopyralid. Above ground biomass, you see a decrease in with application of herbicides, but when you compare sulfosulfuron with the control or untreated plants, they're not significantly different from each other, while these 3 herbicides here were significantly lower compared to the untreated in terms of above ground biomass. So based on data that was collected from this study, we ranked the herbicides according to most injurious to least injurious. So the least injurious was triclopyr, followed by aminopyralid, and then PowerZone, and then the least injurious was sulfosulfuron. So based on this study, we can use sulfosulfuron for sedge control, and then for triclopyr and PowerZone, we should only spray it on the weeds and not on the grass because it can injure it badly. For this experiment, aminopyralid, although we over-applied aminopyralid, it seems promising, but we need further studies in order to confirm this. So now we know now we fill the blanks, all of the blanks here. I'll give you a short summary of what I've done. So for Fimbristylis cymosa, for the propagation portion, you can do hydroseeding. For Sporobolus Virginicus, you can do hydromulched capping of the stem cuttings, but it's best to use apical cuttings which are treated with auxin for 24 hours. For Fimbristylis cymosa, weed control, you can use Oxadiazon for plugs, transplanted plugs, but not for seedlings. You can control grassy weeds with Fluazifop-p-butyl, and then broadleaf weeds for with aminopyralid. You can use it as early as 28 days after sowing or hydroseeding. Sulfosulfuron and power zone are injurious to Fimbristylis cymosa, so you need to apply it as a directed spray. Sporobolus Virginicus, you can use Oxadiazon and oxyfluorfen for transplanted plugs. And for post-emergence control of sedges, you can safely use sulfosulfuron. And then triclopyr and power zone were injurious to the plant, so you should only spray it on the weeds and avoid spraying it on the plant. Aminopyralid, although we have an over-application, it seems promising, but further studies need to be conducted to confirm this. So, future studies for this research. We have nurse crop for direct seeded Fimbristylis cymosa. We have seeding rates and timing of removal. Since during the first 2 months there wasn't any cover generated with Fimbristylis cymosa, we wanted to have a quick cover by using some nurse crops. And then after a few months, we'll remove it with herbicides. And then another future study would be pre-emergence herbicide timing for direct seeded F. cymosa greater than 42 days after hydroseeding. Because based on the previous study, we can't apply pre-emergence less than 42 days after hydroseeding. And then the next one is weed control in hydrocap Sporobolus cutting. So we have how early can you apply pre and post-emergence to control weeds. And then finally, mixed native species hydroseeding, because we want to integrate as many or as much native species as possible in the roadside. So this is another area of further research. So I'd like to thank my committee members, my advisor, Dr. DeFrank, Dr. Kiley, and Dr. Drake for reviewing my manuscripts, and Chris Dacos of HDOT for providing funding, and Glenn Sakamoto and the staff at NRCS for providing us technical support and planting materials for this study. Craig, Ronald, Neil, and Scott for helping out in our experiments. Roger Corrales and the guys at the Manoa Research Station for helping us set up field experiments there. Melinda for helping out in planting and data collection. And the student assistants that helped me in the past 3 years, Tamara, Zach, Mark, and Dave. And I'd also like to thank my family for giving me support and encouraging me to do graduate studies. Yeah. So any questions?
Okay, I think we have time for a few questions, although I think Orville covered almost every single aspect you could possibly imagine. But I'm sure there's probably a few that he may have glossed over. If you have any questions now, we can take it away.
Orville, you described how triclopyr was injurious to Sporobolus monocot, and I'm most familiar with triclopyr being a broadleaf specific herbicide. Do you have any insight to why you would see injury on the grass?
So the question was why triclopyr is injurious to the grass.
Correct.
Whereas it was broadleaf herbicide, specifically for broadleaf. I think it's because I would say it's not exactly because of the active ingredient. Maybe it's because of the concentration. I'm not sure, but yeah.
I can make a comment on that. I think the rates on triclopyr you values were probably 2 to 4x higher than
Okay.
you would normally see in a ranged land situation. The product is labeled under a different name for turf grass, but it's safe on cool season species, fescues and rye, whereas C4 type of grasses are not usually safe. Bermuda grass, zoysia, seashore paspalum. Is ackee grass a C4 plant?
I think it's a C4. Not sure.
It's warm season
Yeah.
grass.
Pretty good chance it's
Pretty good
C4.
chance.
And something in the way the leaves displayed and transport
Correct.
seems to be at the root of that. It's a great study. I mean, really a good study. Even the overdoses. Because you learn a lot from that. You think that's going to happen when landscape crews get out there? It happens all the time.
Any other questions? Okay then, well, thank you very much for coming.